we're loading up the presentation. So the industry mentors, Emma Chory and Richard Schoenfeld. The title of their design project, Sf9-AAV2/HIV. Does everyone get that? I got it. And it's Katie, Caleb, Jaimie, and Paul who will be presenting this design presentation. Yeah.
Is there a clicker?
We don't have one. Okay. Yeah. All right. We'll have a manual clicker, I guess. All right.
Good afternoon and welcome to our presentation. My name is Jaimie, this is Katie, Caleb, and PJ. Together we comprise JPKC Immuno, and we are looking to develop and produce a novel HIV vaccine entitled Sf9-AAV2 HIV in Sub-Saharan Africa. We'd like to start off by thanking our mentors, Ms. Emma Chory and Mr. Richard Schoenfeld. Our mission is to produce a novel HIV vaccine to the region of Sub-Saharan Africa. We would like our vaccine to have a 90% efficacy, which is the standard of current vaccines on the market. Our target population is the number of uninfected individuals in Sub-Saharan Africa, which comprises 947.6 million individuals. HIV is a virus that is spread via contact of bodily fluids between an uninfected and an infected individual. It may be spread via blood, semen, vaginal or anal fluids, or breast milk, for example. The most common way to contract HIV is via anal or vaginal sex without a condom. Once an individual has contracted HIV, they typically come down with acquired immunodeficiency syndrome, or AIDS, within 10 to 15 years. Once an individual has developed AIDS, the lifespan is typically 1 to 3 years. How does HIV work? It infects the helper T or CD4 cells of the human immune system, thus compromising the human immune system. It binds to host cells and it inserts its RNA and then reverse transcribes its RNA into the host cell's DNA. It then replicates and destroys the host cell and travels to other cells within the body. Once an individual's CD4 cell count is below 200 cells per milliliter, they are said to have AIDS, leaving them highly susceptible to other opportunistic diseases and illnesses. Our target population is the uninfected individuals in Sub-Saharan Africa. The reason we have chosen this region is because 38 of the 43 countries identified as extreme or high risk to HIV infection are located in this region. The population is also expected to double by 2050. There are poor healthcare systems, relatively low economic capacity, and relatively underdeveloped countries within this region. For a population breakdown, the total sub-Saharan population in Africa is 973.4 million. There are currently 25.8 million individuals infected with AIDS, which leaves 947.6 million individuals that are uninfected. This leaves 94.76 per million per year, spread across 10 years. There have been several HIV vaccine trial efforts to date, although it is the vaccine that has been studied for the longest time without actually having a successful candidate yet. HIV was identified as the cause of AIDS in 1984, and 4 years later the first HIV vaccine trial was launched. It incorporated the GP120 envelope protein, which is required for HIV to infect host cells. However, it proved not to be very effective. The most successful HIV vaccine trial to date is the RV144 TIE trial, the results of which were published in 2009. This consisted of 4 primer shots called the LVAC HIV, which was a canary pox vector, and 2 booster shots, which is the HVAX BE. The problem was that the efficacy was relatively small, and it decreased by about 60% within just 1 year.
So our technology is going to modify the LVAC vaccine that was part of RV144 while working with Global Solutions for Infectious Disease, who manufacture the AIDS vax, to supply them together in a similar fashion to the RV144, but through our modifications improving efficacy. Sanofi Pasteur owns the intellectual property on LVAC, but they only own it on the canary pox vector, the manufacturing process for that vector. And the genes that are actually delivered that evoke the immune response are not covered. So we can get around this intellectual property by changing the vector. So our vector is a recombinant adeno-associated virus, specifically the serotype AAV2. Recently at Stanford, a paper had been published that mapped out the areas of most significance in which this vector binds to human cells, which could then be taken advantage of to optimize the viral surface to increase binding to cells for drug delivery. It'll contain the same 3 genes, N, GAG, and PRO, which code for proteins on the HIV surface, allowing the body to develop a recognition system for these and have an immune response to HIV. We'll also include the rep and cap genes, which do not have any part to do with the immune response, but they simplify transfection to 1 step. So we're going to be producing them in fall armyworm ovary cells, or Sf9 cells. Most common practice for producing our AAVs is using HEK cells, or human embryonic kidney cells. There's issues of scalability and disposable reactor processing with HEK cells, as well as risks of infection and contamination. Additionally, Sf9 cells have been shown to have greater viable cell densities and specific productivity than HEK cells. So for our manufacturing requirements, we'll need to make 7 × 10⁵ PFUs per dose, which is the basis that the RV144 used for delivering the same genes. It'll be administered in 4 doses for each person a year. And because of the number of around 95 million, we'll need to manufacture about 2.65 × 10⁴ plaque-forming units per year of our virus. So our manufacturing layout starts with a cell culture process where we actually produce the viral vector. This moves into a downstream purification process and then is validated by quality control to ensure that our product is safe to use. So in the upstream process, we're using a 17-day disposable reactor train. Using disposables allows us to eliminate clean in place and sterilization in place, allowing us to turn over batches quickly. This also reduces our capital investment initially, but it increases our consumables costs. And it allows us to build our facility faster and that we have less complicated equipment to work with because the bags already come validated. We apply the worst case scenarios from literature on growth-specific productivity and oxygen consumption in our model. And our vessels will all require dissolved oxygen control as well as temperature control. Dissolved oxygen is monitored via electrochemical probes that are installed in the bags and monitored by the control systems for those bag technologies. The cell line that we're going to be used will be created by Lonza Biologics. As a result, we'll have to pay them 5% royalties each year on what we sell. So the basic process, we start at our vial thaw. We move into our seed reactor train where we're scaling up our culture from a vial all the way to be able to seed our 250-liter bioreactor where we actually produce our product for a 6-day batch. So this is a flow diagram of our upstream process starting from the vial, which is then transferred into our seed train from 2-liter through 100-liter bioreactors. At each stage, adding media to bring our seed density back down to 1 × 10⁶ viable cells per mil. So that way the waste concentrations will remain low and we're keeping our cells in exponential growth. Each stage has air and oxygen supply and headspace, so that way we can have gas transfer. In the stirred stages, oxygen is supplied through a drilled hole sparger because the volumes are too high for simply headspace mass transfer. Of note is that in the 250-liter bioreactor, our production reactor, on day 6 of our production culture, we'll have 225 liters of culture at a final titer of 1.404 × 10⁻14 plaque-forming units.
So from the upstream manufacturing facility, we'll move into the downstream purification scheme. Current purification steps usually have multiple columns. However, our process will use a single heparin column with a very high affinity for AAV2. The purity level of this heparin column is around 90%. This purity level is the number of empty particles to the AAV particles. You want to have a higher purity to have a higher concentration of AAV in your final product coming out of the heparin column. Another step is the polishing step. We'll be using a size exclusion chromatography column for our polishing step. So moving into the process flow diagram, the solution will come in at 225 liters and enter a centrifuge. This is the biggest step in the purification process. This will reduce the 225 liters around tenfold to 20.5 liters. This is a much smaller volume, allowing for smaller columns, a smaller purification scheme, and ultimately reduced costs. So when the centrifuge reduces down the volume, we'll have a cell pellet that will be resuspended in a buffer solution of sodium phosphate and sodium chloride. This will then enter a microfluidizer. This microfluidizer shears the cell membrane, releasing the AAV2 into the solution. Also, when it releases the AAV2, DNA enters the cell lysate, and we do not want this DNA fragments in the solution. So we'll be adding a benzoate nuclease, and this needs to be incubated at 37 degrees Celsius for 1 hour. From this, we enter two filtration schemes, a 0.25 micron filter and a 0.2 micron filter. This is unusual for a purification scheme, the 0.2 micron filter. This is due to the small size of our particle. It's about 20 nanometers in diameter. In comparison, the adenovirus is 80 nanometers in diameter. So these two filtration steps will get rid of the adenovirus, leaving only the adeno-associated virus. We'll then be moving into a heparin perfusion column. This will have the heparin molecule bound to beads in the column, and when the AAV2 flows through the column, the AAV2 will bind to the heparin and everything else will flow through the column. We'll then have multiple wash steps, which will wash out any contaminants and leaving the AAV2 bound to the heparin in the column. To get the AAV2 out of the column, we'll need an elution step. We'll be eluting the AAV2 with increasing concentrations of sodium chloride. The sodium chloride will release the AAV2 from the heparin, and this will flow out of the column. One big advantage of this column is it's a perfusion column. This means the beads have porous flow channels, so this solution can flow through the column in faster flow rates and also increase the surface area, so you have a higher AAV2 binding to heparin. We'll then move into our size exclusion chromatography column. This is the polishing step. So this means after this, we'll have a very high concentration of our product and very few contaminants. AAV2 has a size of 600 kilodaltons, while some other contaminants have a size around 200 kilodaltons. So the AAV2 will flow through the column while other contaminants are trapped in the pores and the beads. We then move into our final formulation where the sodium chloride will be replaced with a PBS buffer, and this will be going into vials. So overall, our purification step, the incoming material is 225 liters at 1.4 times 10 to the 14th PFUs. Our purification scheme will reduce this down to 150 milliliters at 8.81 times 10 to the 13th PFUs. Overall, this is a loss of 66% in PFUs. While this may seem really high for a purification unit, current methods are around 50 to 60% loss. The reason our loss is a little bit higher is AAV2 purification schemes are relatively new. There are very few places that have large-scale manufacturing plants, and the technology is not where it should be and is not as advanced as other purification technologies. So we are going to look to improve our purification by improving the flow rates, the pressures, and continuously improving the single-use technologies as they develop. From the purification scheme, we'll have 125 million vials. This is higher than the 95 million that we were targeting. However, this is due to the bags we'll be using in cell culture. They come in standardized bags, and customized bags will be more expensive. So we will have a little bit higher than our target production. However, this could be due to losses in distribution, and it's never bad to have more than what you need. So before we release the vials to market, it needs to go through quality control. We need to make sure that our vaccine is up to date with our standards. So we'll be using an HPLC to analyze the sample size of our batch. We'll be using a non-porous 2.5 micron methacrylate resin and a TSK gel. The assay time will take around 20 minutes, and this is a vast improvement over other methods which use a 72-hour process. We'll be measuring the particle to infectious ratio, or the P to I ratio, which measures the particle and any infectious materials such as viral contaminants. We will need to develop an SOP to determine what our P to I ratio will be, and any batch that leaves our facility will need to be up to this standard.
For the location of our manufacturing plant, we have decided upon Durham, North Carolina, because it's a central hub of several different institutions and industrial powerhouses in vaccine production, such as GlaxoSmithKline, Pfizer, Merck, Duke University, Wake Forest University, and UNC Chapel Hill. Duke University in particular has a unit that is specifically dedicated to HIV vaccine research. This location will give us a lot of technical and managerial talent in the realm of vaccine production. Furthermore, the utility costs and tax rates in this region are relatively low. There are several safety considerations to take into account with our process. To this end, a thorough HAZOP analysis has been conducted on each unit within the upstream, downstream, and quality assurance portions of the process. Another important factor to consider is the fact that we're using recombinant adenoviral vectors as opposed to adenoviruses themselves. Adenoviruses are associated with respiratory infections such as the common cold, pneumonia, and bronchitis. They're stable under even extreme conditions, and they can be transferred via direct contact such as a handshake or inhalation. They have been assigned by the Occupational Safety and Health Administration, OSHA, to biosafety level 2, which contains agents that pose a moderate threat to personnel in the environment. In contrast, recombinant adeno-associated vectors have been rendered replication deficient via genetic engineering. They no longer contain the structural genes necessary for replication. They are non-pathogenic in humans and animals and have been assigned by OSHA to biosafety level 1. We also must consider the safety of administering the vaccines to patients. First, the booster shot contains alum adjuvants that are regulated and maintained at FDA-regulated levels. Common side effects of vaccines must also be taken into account. These could be irritations or rashes at the injection site, or syncope or fainting due to decreased blood flow to the brain. This presents a risk of injurious falls post-vaccination, and thus patients should be given a snack or a beverage and asked to lie down for about 15 minutes after they are vaccinated. Some patients would have to be excluded from vaccination based on their medical history or current condition. In order to help regulate the safety of our vaccine, there is no such foundation as the FDA within Sub-Saharan Africa. Thus, we must work with the World Health Organization subgroup, the African Vaccine Regulatory Forum, which also works alongside the Program for Appropriate Technology in Health, or PATH, which is based in 70 countries throughout Africa. Together, these two organizations have helped to implement both the clinical trials and the administration of 2 other vaccines for meningitis in 25 countries and malaria in 7 countries. With regards to the attitude towards gene therapy in general in Sub-Saharan Africa, the population is relatively hesitant towards genetically modified organisms, which have recently been proposed as a way to increase the food supply. However, we believe that the attitude toward vaccines may be a bit different because of the recent success of other vaccines. Furthermore, there are several institutions in South Africa alone, such as the University of Witwatersrand and the University of Cape Town, that are actively involved in antiviral gene therapy research.
All right, so now that we kind of introduced what our vector technology is, talked about our manufacturing process, and kind of addressed the issues that surround HIV in general, we're going to get into some of the economics of our company and kind of introduce some of the numbers surrounding building our manufacturing plant, surrounding the cost of production, and we're also going to look to specify some of our economic decision making and kind of explain our reasoning for some of the economic analysis. So first, a quick market breakdown. So as we said, we're targeting 95 million individuals annually. And the total population in Sub-Saharan Africa is expected to double by 2050. So if we assume that this is just a linear increase in population from now until then that means in 2027, when we plan to first reach the market, after assuming a 10-year time of startup and clinical trials, then that means that this 95 million target is only 7% of the total Sub-Saharan population. So while 95 million individuals is a very large market, we believe that because it's such a low percentage of the total population, that in our initial years in the market, it's a number that is achievable. Here we see just a quick breakdown of our revenue. So targeting 95 million people each year at a cost of $4 per person. This equates to about in the ballpark of $380 million in annual revenue. So that number does seem high, but when you compare it to other vaccines such as the HPV vaccine, which nets in about $1.2 billion in sales. Something to consider is that other vaccines are often offered in higher profitability markets such as the US, whereas we're only targeting Sub-Saharan Africa. However, our market will receive less revenue, but our total population that we're targeting is very large. So we do believe that this revenue is achievable. And another thing to note, the price of $4, we chose this to try to minimize the cost so that the people in sub-Saharan Africa could afford it. And we also chose it so that we could still attract investors to our business. So the first major hurdle that we're going to have to overcome is covering the cost or the price of the vaccine itself. Because we're in sub-Saharan Africa, most individuals and many of the governments can't actually afford the vaccine, even though it's only $4. So we recognize that we're going to have to build relationships with people who will be willing to sponsor the vaccine once we reach the market. So in fact, the good news is that these sources of funding are out there. So around $20 billion is spent globally, just in low and middle income countries alone, on HIV efforts. And we have identified 4 different sources, or the 4 major sources for this total funding that we want to try to pair up with so that we can cover our vaccine. So the first are the domestic sources within sub-Saharan Africa. These are just the governments within there. While most of them can afford it, some of them can, and of the ones that can, they have made significant efforts to provide lifelong treatment for HIV. So we're hoping that when a vaccine comes around, if we're able to achieve it, then they'll continue to pursue these efforts. The second major source are donor governments. So this includes the US, the UK, France, the Netherlands, Japan. All of these countries combine for more than $8 billion spent annually on HIV efforts. And then the last 2 are multilateral organizations such as the World Bank and the Global Fund, as well as philanthropic funding. And these sources are more sources that are looking to sponsor the vaccine just because they want to help out and for the greater good. And together these combine for more than $4 billion spent annually. So we're hoping that from this total pool of $20 billion spent on HIV treatment, efforts, research, that we can pool together the $380 million that's needed to cover the price of our vaccine. So next we're going to talk about the funding. This is different from sponsorship in the sense that funding is, we're talking about people who are investing in our company before we're reaching the market. So while we're in early stages and trying to build our company. So we broke our project timeline into 20 years. We're assuming that it's going to take us 10 years of clinical trials and startup and other things associated there to reach the market. Then we're hoping to reach our target population over a 10-year span. And we broke this down into 5 different stages. The first stage is our initial R&D. That's where we're going to try to be improving the science of our vaccine. The second stage is going to be building our manufacturing plant. We're hoping to do this after 2 years, which is relatively early. However, the reason we want to do it so early is so that by the time we're in clinical trials, we have a fully functioning facility that we can operate in. And then stage 3 is going to be the heftiest amount of work, requiring about 6 years, and this is going to be all of the clinical trial phases. That number of years can go up or down, but that's the estimate that we have now. And then stages 4 and 5 are when we reach the market, and then the 10 years that we're trying to reach the people that we're targeting. So now that we kind of outlined the timeline of our project, we identified the types of sources of funding that we want to receive in each stage of the project. Here we have listed specific organizations and companies that we're trying to target, but I wanted just to cover the reason why we selected these types of sources. So first in stage 1, the initial R&D phase, we're going to primarily seek grants, and the reason is because we're not going to be able to afford to acquire debt early on in our business life, so we're trying to avoid that as much as possible. Another source of funding will be issuing preferred stock so that we can try to attract venture capitalists and angel investors to invest in our company if they like our idea. In the second stage of building our manufacturing plant, the biggest thing is having liquidity in our business and a lot of cash so that we can build this plant within 2 years. So because of that, we're going to seek to issue some bonds and try to seek bridge loans from banks so we can acquire that cash to build the manufacturing plant. And this is when we're going to really start acquiring debt in our business. In stage 3, all of the clinical trials, we estimated that this will be around $500 million of a total cost. Again, that could be more or less. But now that we're dealing with a much more significant numbers to fund our business, we're going to start seeking money from those donor governments who might have higher pools of money and who are willing to help us out. And then lastly, in stage 4 and 5, this is when we're generating revenue from our vaccine, so that's going to be our major funding source. And as we continue to operate, we might buy back some of the bonds and some of the stocks that we issued out so that we can reduce our dividend payments and also reduce the interest that we're paying as we operate. So now that we kind of talked about the qualitative stuff, here are some of the numbers that surround our building our manufacturing plant and our production. So first, we estimated that our total cost to build our manufacturing plant is around $15 million. This number is actually very low, what you would compare to most biological plants that can run upwards of hundreds of $millions. This is an initial estimate, and the reason that it's very low is because our entire process is a single-use system, so the initial investment into equipment and infrastructure might be lower than normal, and also because we're only operating 4 batches each year. Each batch produces a significant amount of our product, so we actually don't need a very large facility. And here are our costs of production. We estimated them to be around $12 million in total. The bulk of these costs of production are actually going to come from paying interest on the fixed capital and also paying the royalty to Lonza to use their cell line bank. Something to note here is that $12 million is relatively or very low compared to our ballpark estimate of around $308 million in revenue. However, in the next slide, we're going to talk about where the major expenses of our business will come from outside of the cost of production. So because it's going to take us 10 years to reach the market, we're going to acquire a significant amount of debt in that time leading up to. And so a lot of our expenses as we're operating are going to be paying back that debt. So we estimated around $80.5 million will be spent there annually. Then we also know that we're going to acquire a significant amount of cost to reach our target market. There might not be infrastructure in place or administration systems to be able to reach these countries and to reach these individuals. So setting up those distribution channels is going to be a hefty task as well, and we predict that this will be around $100 million annually. So with all that being considered, we put together kind of a final valuation of what we see our business at. So we estimated that after 10 years reaching our entire market, our business would be around a net present value of $220 million. When you compare this to most biopharmaceutical companies, especially large ones, it's relatively small. However, we do think it's large enough to continue to attract investors. And then the biggest note here is that the value of our business would actually reach a peak within the middle years of production. And this is because we're going to be offering our vaccine at a constant price throughout 10 years, whereas most drugs or most goods and services, the price of their price will actually increase just due to inflation and due to the time value of money. However, we're going to offer it at a constant rate, and we're going to continue to operate even though the value of our business or the net present value of our business is decreasing. And this is because our mission is to reach our target population, not necessarily to make as much money as we can.
So to review, ChE Immuno is going to offer our vaccine at $4 per person. This is lower than a lot of the other vaccines offered, like such as hepatitis A, which is offered at $18, and hepatitis B, which is offered at $12. However, we will be able to make profitability due to our low manufacturing costs due to single-use technology. Our novel AAV vector allows for smaller batches with higher titer, and our one-column purification scheme, which allows for faster purification and less cost. We also have a large target market at 94.76 million people annually, and we'll be generating an annual revenue of $379 million. While this does not seem like a lot compared to other drugs, and while vaccines are not the blockbuster drugs that big pharma companies are looking for, there's still many reasons and incentives to produce vaccines. The first is fully immunizing babies results in $10 saved for every dollar spent. The second, in regions where there are poor healthcare systems, lack of administrating doctors, and people with low incomes, a preventative vaccine is more profitable for big companies rather than a treatment solution. In conclusion, Sf9-AAV2 HIV is a low-cost, profitable vaccine that will reduce the number of individuals with HIV and prevent millions of deaths from AIDS each year. Thank you, and any questions?
presentation. Have you considered the high viscosity that you're likely to have when you have your AAV very concentrated? How are you going to do the liquid handling, filtration, and so on?
Our column will take a long time. I think we've calculated to be about 4 hours to run through 300 mils through our column. I guess with that high viscosity, we're going to have to allow for a long time for that column to work.
And I didn't realize that you only run 4 batches a year. The batch time from beginning to end is not that long. We're talking a month. What are you going to do in the rest of the year between batches?
Well, in between batches, batch records need to be prepared for the next batch as well as validated by regulatory agencies. And we're also going to have to validate the previous batch record to assure that the last batch is safe for distribution. So after the batch ends, we'll actually have to take time to look through all that paperwork and submit it to make sure we can actually distribute what we have.
That's all very true. Ordinarily, companies use separate staffs to do the review rather than the same staff.
Yeah.
If you use the same staff, there's some ethical issues that come up. Can quality people be the same as the manufacturing people because they're doing the checks on themselves?
Yeah. The extra time also gives us time when we set up our SOPs and develop process characterization saying whether a batch passes or fails. It gives us extra time to be able to turn around and make sure we hit our supply if we do need to run again, so we have a loose schedule to be able to supply our market.
Maybe the right answer would be we've left ourselves room to service an increasing market.
Yeah.
So I like the slide, Jaimie, I think it was you, about the societal, ethical impacts, especially putting it in perspective of sub-Saharan Africa. But as you know, in the US, there's been an incredible debate about vaccines that just doesn't seem to be going away, despite my puzzlement over that debate on vaccines. But you didn't touch upon that ethical concern or that societal concern with vaccines. I'm wondering if you kind of understand that issue of the public debate in vaccines in the US and how your approach would either add fuel to the people against vaccines, or would it even encompass that debate at all? And you don't have to, just because you talked about it, you don't have to answer it, but that's to everybody.
Okay. I think that it probably would add fuel to the debate here in the US, because people may look at our organization and say, okay, we're just going ahead and implementing this in sub-Saharan Africa, assuming that they are okay with it and that they would be in support of this sort of strategy. I think that, as you said, it's an ongoing debate, so it's something that the world is going to be continuing to consider whether or not we go ahead and implement this strategy. And it's just, we will have to make sure that we explain this properly to the population that we are administering it to, and I think education is going to be key. And then that way there might be less criticism on the end of the US.
Has anybody seen the new movie? I have not.
I haven't heard about it.
What movie is this?
The anti-vaxxers, I don't know who sponsored the movie, but it's going to do a terrible disservice to.
Could it be Michael Moore by any chance?
You never know. Is it?
I don't know.
Something like that.
Might be Jenny McCarthy. She's a big one on that.
Yeah, the issue is thimerosal and the mercury, and people say, well, some people say it causes autism. But this movie is going to go and discuss that and show that it's going to be on the side of the anti-vaxxers. And it's going to make it worse. I think education is the right answer. All of the scientific evidence that's put forward is saying it's absolutely safe. It doesn't associate with no correlation.
And that was actually my question was related to the mercury, right? Because the people who are against
It's
vaccines,
still the best preservative, right?
right? And I don't know if you were planning on using mercury. You know, you mentioned alum adjuvant, right? But as far as I know, the debate is not related to the alum adjuvant, it's related to the mercury that is used, as Richard was saying.
There's more
So-
mercury in your body from the fillings in your teeth
Right.
than you get from the vaccine.
My other question was, I like how you're exploring a different cell, right? And understanding that there are problems, with current cell lines that are used, and you would like to look at your SF9?
Yeah.
SF9 cells. But there's a disadvantage to that too, right? Whenever you try to go against conventional
Yeah.
wisdom. So I wonder if you could comment on your
Sure.
thought processes.
Yeah, so going from HEK to SF9, so I've had experience working with HEK, and you can actually work with them in fed batch processes.
Why don't you explain what HEK stands for? People
So,
will get
yeah,
the message.
they're human embryonic kidney cells. Because they're human-based cells for producing drugs, they're very sensitive in terms of mammalian cells. Human cells require a lot of different controls and production processes, like there's issues in maintaining carbon dioxide at a certain level, oxygen at a very certain level. They need a very balanced diet of nutrient feed daily that it's kind of hard to pin down exactly what it is to get them to produce in a robust manner. So you could run the same batch twice and get very drastically different results. So SF9 cells, what we were able to find in the literature is just batch processes, not fed batch, that produce enough titer to supply our market. So that eliminates those complications, but it also leaves us with the opening to contribute back to the scientific community later. And as we grow and maybe try and explore other markets, we can improve with possibly a perfusion-based seed train or a fed batch to try and increase that titer and growth.
The other major advantage of SF9 is the HEK cells. We've got billions and billions, trillions of amplifiers for human diseases. So people shy away from using them whenever possible. We'd rather use an insect cell, a hamster cell,
Yeah.
something that is not likely to carry human viruses.
Also, HEKs can be grown in suspension, so it makes the production scale up almost impossible.
Good point. Good.
I have a question. Is there, given that the virus vector that you're using has canonically been used in gene therapies, is there anything in your research that you perhaps learned that you could translate in what lessons have been learned in gene therapy and apply it to vaccinations, given that they're almost entirely different fields but the same product?
I mean, you're supplying genes to a cell, so the delivery mechanism is the same. You would just be changing what's inside, so you for the delivery method. So you could probably use a very similar process, like pretty much exactly the same, but just change your actual target for what you're doing to inject into the human cells to produce what you want them to. So for like example, if somebody had some sort of genetic illness and they couldn't produce some sort of protein, you'd be able to deliver that kind of gene to them in order to be able to get them to make that using this kind of vector.
Good. Any last questions?
No, I have a comment.
Yes.
Is this the first time you've met Emma?
In person, yes.
Yeah.
Yeah.
It is not the first time you've seen her, however.
No.
So you know that?
Yeah.
What? I know, it's like
They're up, they're still up. All the posters
oh,
are
the
still up.
posters? No, they took them down yesterday.
Well, they're perhaps yesterday.
Yeah, she was just telling us
They
about
They just took them
that.
down.
What is it?
Yes.
She had
Yeah.
on the
One
banner
of the poster
or
children.
whatever.
The poster.
She is,
I used to be
yeah.
on the poster.
I didn't recognize it though when we first saw you on
Yeah,
Skype.
they just
It wasn't
took
until
them over.
today.
It's been too long.
Well, we should give you those.
My mom wants one, so I'm gonna like head
We'll have
over
to
to
track
alumni
that down.
relations and see. I don't know.
All right, excellent. Thank you, guys. That was great.